Hello everyone, welcome to today's webinar featuring a discussion with Professor Travis Craddock on the study Genetic Predisposition for Immune System, Hormone and Metabolic Dysfunction in ME/CFS that he co-authored as part of a special ME/CFS themed issue in the journal Frontiers. Some of you may remember that we had, this is a series, we're on part 2 of a 4-part series, and we kicked off a couple of weeks ago with Ken Friedman. So this is the number 2 in that series. Professor Craddock we're thrilled to be joined by him today, and I will give you a little bit of information about his background. Sorry, the slide's not coming up. Okay, so Professor Craddock is an associate professor in the Department of Psychology and Neuroscience, Computer Science and Clinical Immunology at Southeastern University in Fort Lauderdale, Florida. He serves as a director of Clinical Systems Biology Group at NSU's Institute for Neuroimmune Medicine. He researches complex chronic illness involving neuroinflammation, including ME/CFS and Gulf War illness. And this year alone, he's published 5 papers related to ME/CFS, putting forward hypotheses, commentaries, and findings from original research. Travis received his PhD in the field of biophysics at the University of Alberta. So thanks to everybody for joining, and I will hand it over now to Professor Craddock.
Hello, everyone. Thanks for having me. I'm just waiting for my slides to appear here. Okay, excellent. So yes, today I'm going to be talking about genetic predisposition in ME/CFS. I'm actually going to be talking overall generally about our genomic work here at the Institute for Neuroimmune Medicine. So I'll be talking about 2 papers with the ending focusing on this genetic predisposition. The 2 tend to play off one another, as you'll see here in a moment. So this was work that was done by people in my lab, students Mary Jeffrey and Melanie Perez, working in conjunction with Dr. Lubov Nathanson and Nancy Klimas, who are also here at NSU's Institute for Neuroimmune Medicine. Next slide, please. So first off, I'd like to thank the Solve ME/CFS initiative for hosting these webinars. I think it's really great to get this information out to a wider audience. We tend to publish as much as we can on the work that we do, some of our ideas, thoughts, and processes, as well as our research findings. But getting a platform to get it out to interested people, other researchers, the interested patient community is a great help. I mean, giving this illness more exposure to let people know the findings, the good research that's being done in this area is always a good thing. Next, please. Okay, so first off, before I get into talking about the research itself, I would like to thank the groups that have provided funding to perform this research. Now, as was mentioned earlier, the type of research that I do straddles both ME/CFS as well as the related sister illness, Gulf War illness. My work is primarily in Gulf War illness, so a lot of my funding comes from the Department of Defense through the congressionally directed medical research program. And the Department of Veterans Affairs, specifically the Miami VA healthcare system. This work in Gulf War illness is very applicable to ME/CFS, and so what we tend to do is build platforms and algorithmic processes to examine Gulf War illness, but then transfer it over to our same research studies in ME/CFS. Now, research for us in ME/CFS has primarily been funded by the National Institutes of Health. And because we're a computational group here, all of our computational work is done down at the University of Miami's Center for Computational Science's high performance computing platform known as Pegasus. Now, these awards that I'm talking about are not primarily to myself. There's a couple there that are to me, but they've been awarded to many people in our group, including Nancy Klimas, our former group member, Dr. Gordon Broderick, Marianne Fletcher, and Lubob Nathanson. Next, please. Now, I'm sure that most people are familiar with this, but I always like to start off a presentation for those that may not be as familiar with the disease as some of us that are in the field are. So myalgic encephalomyelitis, ME, or chronic fatigue syndrome, which is a related, closely overlapping illness, CFS, is a chronic multi-symptom disorder. It's primarily characterized by persistent fatigue lasting more than 6 months, which is not alleviated by rest. And this is usually accompanied by a constellation of other symptoms, ranging from gastrointestinal problems, muscular and joint pain, to working memory problems and brain fog. It's been estimated that this affects somewhere between 400,000 to 900,000 Americans, or I mean, more recent estimates I think might be over 1 million. These are older numbers, which I believe has been quoted as more than the individuals affected by Parkinson's disease and multiple sclerosis combined. But again, I'm quoting off of numbers that I can't quite recall off the top of my head here. Now, it's also been estimated that this illness affecting so many people has resulted in approximately $9 billion in lost productivity to the U.S. annually. So this is not a minor or negligible problem. This is a very large problem that we should be looking at. We should be trying to find illness or treatments for this illness, but no treatments have been found to date. It's primarily diagnosed on symptom burden, and we're looking for biological diagnostics to nail down objective biological ways to diagnose the illness. And once we do that, we should be able to try and pinpoint treatment avenues as well. So this illness is affecting a lot of people. It's very debilitating, and progress needs to be made. Flat out, it just needs to be done. Next slide, please. So about 6 or 7 years ago, Dr. Nancy Klimas, who I don't think needs an introduction here, but who's a leader in ME/CFS research, she moved from the University of Miami up to Southeastern University here in Fort Lauderdale, Florida. And she was given the opportunity to start up this brainchild of hers called the Institute for Neuroimmune Medicine. And so since that time, she's been working on this mission to advance knowledge and care for people with complex neuroinflammatory illnesses through integrating research, clinical care, and education. And so that's neuroinflammatory illnesses, complex chronic conditions, that encompasses ME/CFS as the prime illness that we research, as well as Gulf War illness because it's a closely related sister illness. Next slide, please. Now, the way that the Institute for Neuroimmune Medicine, or INIM, is set up is kind of how it's drawn here. Is in the center, we have the director, Nancy Klimas, who runs the clinic, the administration center. And then there's 4 main research cores surrounding that prime clinic that provides care for individuals with ME/CFS. So first, there's the clinical research group. This is directed by Elizabeth Balvin. And this is where individuals from the clinic are drawn into research studies. These can range from nutrition studies to genomic studies to exercise physiology studies. Basically, everything that we dream up goes through this clinical research program. Now, during the clinical research, this is where lab samples are drawn, and from the blood and bodily samples that are gained from this clinical research, it's handed over to the Immunology Genomics Lab research core, which is directed by Marianne Fletcher, Dr. Marianne Fletcher, and the genomics group is headed up by Dr. Lubob Nathanson. And this is where they run full-scale analyses of any sort of biological measure you can dream up of. So cytokine panels, cell counts, cell activity, RNA-seq, microarray expression, cell energetics, bioenergetics, that sort of thing. That all goes through that lab. Now, because of these high throughput platforms, which measure many biological markers, we have a wealth of data that comes out of that lab. And that data gets then passed to my group, which is the clinical systems biology group. And we take that data and attack it from a twofold perspective. One, we try to analyze the data for patterns to look for things that distinguish illness from control groups. As well, we build theoretical models of how we think the body is supposed to work, and then tune it to the data such that we can produce a virtual model of ME/CFS that we can then computationally test virtual treatments on to try and come up with treatment avenues that are viable for this illness. When we find those viable treatment avenues, we then pass it off to our animal research group, which is headed by Dr. Mariana Morris. And there they can test for safety and efficacy of treatment avenues. And then that gets fed back in. If we find any viable treatment avenues, it goes back into the clinical research group where we actually test out the treatment courses in phase 1 clinical trials. Next slide, please. Now, my research group here, we're a very diverse group. We have people from computer science, from psychology and neuroscience and biology, to clinical science engineers, medical students, and so forth. So we'll take anybody as long as they're interested in working on this problem. So this is primarily my group. Again, this is ranging from undergrads up to graduate students and staff as well. And this is only for the clinical systems biology group. There's a whole host of other individuals that work in the rest of the clinic. So I just wanted to thank these individuals for their time and effort into our research projects. Okay, next slide. Now, the way that our clinical research primarily works is ME/CFS is a fatiguing illness. So we want to look at how an individual responds to a challenge. So if it's a fatiguing illness, we tend to challenge that by an exercise. So we call this a dynamic modeling method, or a dynamic modeling challenge. And what we do is we measure symptom burden prior to exercise. So at baseline, we measure people's symptom burden on many scales, SF-36, MFI, Davidson trauma, that kind of those kind of measures. And then we put the individuals on a bike and we measure their blood. We take blood measurements at many time points over a exercise challenge. And primarily the working paradigm has been one measure at rest, one at peak exercise, and then 4 hours later after peak exercise. This gives us a dynamic description of how someone with ME/CFS responds to a physical challenge. Now, beyond this, we've added more time points to get more resolution in the data, but these 3 time points have been the prime scaffold on which we've built the rest of these dynamic modeling studies on. Next. So like I said, once we've measured these or taken the blood samples from individuals over this exercise challenge, it goes to the lab. Primarily, we do a blood analysis on peripheral blood mononuclear cells, or PBMCs. We'll measure things like hormones, so your stress hormone cortisol, or the sex steroid hormones, estrogen, testosterone, progesterone. We measure things like flow cytometry, so measuring immune cell counting. And then we measure actually not just the number of cells or the types of cells, immune cells that are in the blood, but we also measure how they're talking to one another. So we measure the cytokines, or the signaling molecules that these immune cells use to talk back and forth. Additionally, we can also measure the expression of genes within these cells. And generally, what I'm going to be talking about today has been from these cells overall as a homogeneous group of immune cells, but work done by Dr. Maria Abreu and Lyubov Nathanson here has started to focus in on individual cells, so isolating different subsets of B cells or T cells and looking at how the gene expression in those individual cells is taking place. So getting higher resolution and more fidelity in these studies. Next slide. So again, like I was saying, what we tend to do is here at the Clinical Systems Biology group is we want to take that data from the patient, which is kind of that first image there, construct it into a theoretical model using the data from the patient to try and predict drug treatment courses, and then feed that back in to individuals in clinical research trials. Now, that second stage there where we construct these models or try and analyze the data to come up with a description for how this illness is different from typical healthy controls, there's generally 3 steps that are involved. One, we want to identify putative targets. So what is it that's different in these illnesses or in ME/CFS compared to healthy controls? And we don't want to just look at what's different, but we want to look at what's different and what can we affect with drugs that are already on the market. So we're really looking at a rapid intervention type strategy. We could use the old pharmaceutical strategy where we're coming up with new compounds, but this can take 10, 15 years in order to develop efficacy and safety protocol and guidelines. But we don't want to do that. We want to leverage the safe FDA-approved drugs and nutraceuticals that are already available to try and come up with a treatment now, not 10 or 15 years in the future. So once we identify these targets, we use the models of the system to simulate and optimize a treatment course in which to return ill individuals from their ill state back to a healthy state, or a reset of the system's homeostasis. So return you from an ill state back to typical health. That's the overall arching theme that we work with. And then once we've identified the optimal treatment course, we try and minimize adverse drug effects and off-target interactions to make sure that the drug treatment course is as mild and palatable as possible for these individuals. Okay, next slide, please. So today I'm only going to be talking about step 1, and this is primarily from our genomics work. I'm going to be talking about 2 main papers. Both of them came out this year. One with treatment avenues in ME/CFS, chronic fatigue syndrome, which looks at gene expression data, and then another one which looks at GWAS, or genome-wide association study, to look at are there genetic differences in individuals with ME/CFS compared to healthy controls. So this gene expression study, the first thing we did was we looked at 23 women with ME/CFS, with CFS as defined by the Fukuda definition, and compared them to 15 demographically comparable healthy controls. We used Affymetrix microarray to measure gene expression. And we didn't look at individual genes. What we did was we took that data and looked at known biological pathways or cell signaling pathways, and looked at groups of genes together and looked at how different are those groups of genes compared to how it is in healthy controls. So we're looking at gene pathway differences, not individual genetic or gene expression differences. We looked at which of these gene groups are different in ME/CFS, and then we correlated that with how severe the symptom burden was in individuals. So we looked for those gene pathways that correlated more strongly with certification on higher levels of fatigue or brain fog or cognitive problems, to look for which ones seem to be driving the symptom presentation in individuals. Once we found those and ranked those on their overall effect on an individual, we cross-referenced all the targetable proteins within those pathways with known pharmacogenomic databases to look at which ones of those modules can we actually potentially modulate using a drug treatment course. Next. Okay, I'm actually going to get you to just go to the next slide after this. It's going to be the same with some red blobs on it. Okay, so what we did is we took the top, we looked at a total of 4,620 of these gene pathways or these gene modules. And we looked at roughly the top 1% of the modules that were different in ME/CFS compared to healthy controls. And of those top 50 modules, we found out of the total 8,602 genes that we looked at in this study, that 437 of these genes were differentially expressed compared to healthy control. Now, we took those groups of genes and annotated them using something called overrepresentation analysis to kind of name the groups of genes that we were pulling out. And that's what that network diagram is over there on the right. And I know it's very, very difficult to see. It's very small. There's a lot of detail there. But what I've highlighted are the main areas of interest. So there seems to be these clusters of gene pathways or interacting gene systems and networks that all revolve around a central issue. So the three main areas of interest in this were things like B cell and T cell receptors. So we're talking about adaptive and innate immune cells and how they talk to one another. And then 2' cells or immune signaling molecules, one being tumor necrosis factor alpha, and the other being transforming growth factor beta or TGF beta. Next slide. Now I'm just zooming in here on each of these areas. So if for those individuals that might be interested in the prime pathways that are involved, here we can see, oh, sorry, go back one slide. So here is B cell and T cell receptors. And so that encompasses namely B cell and T cell receptors, but as well as these pathways involved in adaptive immune systems, cell adhesion, RAF-MAP kinase cascades, as well as something which is related to axon guidance. But it may be a misnomer in this case. The reason that is coming up is that this is involving internal cellular architecture with something known as the cell cytoskeleton. That's primarily active in axon guidance, but it's also active in immune cells when they change from being dormant to in a more active state. So you have changes in the way that the cytoskeleton is regulated in that. So that may be a bit of a misnomer. The next slide will show us the same modules that are involved with tumor necrosis factor alpha. And so primarily this is a innate immune system signaling, and it's also involved in apoptosis, as well as various transcription factor networks. And then the next one is transforming growth factor beta. And this overlaps with, so transforming growth factor beta signaling pathway also overlaps with the tacrolimus and cyclosporin pathway, which is in some way related to IL-2 signaling. So there's this kind of overlap in the way that certain cytokines are produced and released, and there's a lot of the machinery that are used for certain the creation of certain signaling molecules is also present in the creation and usage of additional cytokines. So this one primarily seems to focus around TGF beta, but there is a tie-in to additional signaling molecules. If you go to the next slide, this shows the correlation of these modules, these individual blue central modules with the severity symptoms. So this is the B cell and T cell receptor signaling clusters, And we have measures of the SF36 multidimensional fatigue inventory, the Krupp scale, the sickness impact profile, and the Pittsburgh Sleep Quality Index. And we can see that a lot of these things with the module associated with adaptive immune signaling focus or can affect, or it correlates strongly with physical limitations, general fatigue, physical fatigue, and overall total description of the multidimensional fatigue inventory. Whereas things such as B cell receptor signaling, T cell receptor signaling seems to primarily focus on pain in the SF36 scale. So it seems to be this overall the immune system, misregulation in the immune system is giving rise to these physical limitations, but primarily, specifically, the receptor problems or the differences in receptor expression is what's giving rise to something like pain or problems in sleep quality, as you can see by the last column over there. Next slide. So overall, this talk back and forth between B cell and T cell seems to globally encompass the problems that are going on that we see with symptom burden ME/CFS. We get something very similar for TNF alpha signaling. It's quite broad across all of the symptoms. So again, things like the innate immune system, the second last two rows there, is broadly spread across many of the scales for the multidimensional fatigue inventory, as well as individual scales for tumor necrosis factor alpha. So these signaling, the pro-inflammatory cytokine TNF alpha seems to generally give rise to many of the problems that we see in ME/CFS. But if we look at the following scale for transforming growth factor beta, we'll get a different story. I'll just wait for that slide to pop up. I'm looking to go to slide 24. There we go. So here we can see that primarily the TGF-beta signaling pathway is correlated with pain and nothing else. So this gives us kind of helps us narrow in that if we may want to be looking for a specific treatment for pain, we maybe want to be focusing on targeting the TGF-beta signaling pathway. Whereas if we want to broadly affect the illness and cover a large swath of the symptoms that are presented in ME/CFS, we may want to focus in on TNF alpha signaling or the differences that we're finding in T cell or B cell receptor on immune cells. Next slide. Now, if we take those individual genes and we cross-reference them with the drug databases, as it'll be shown on slide 25 coming up.
And Travis, the slides are moving, I think, for everybody else. I think there's a lag on your end. So
okay. I apologize.
The slide, when you say slide, next slide, they are moving. So probably I would just keep going, even if you can't quite.
Yeah, okay, I'll follow along here then. Okay, that's good. So when you cross-reference these individual genes that belong to these pathways with pharmacogenomic databases, we found a total of 242 gene-drug interactions. 92 of these are FDA-approved drugs, and they target only 39 of the roughly 400 genes that had come up. So there are treatable targets in 37 of the 50 gene modules, and of those 37 targetable gene modules, 23 were highly correlated with the symptom burden or the symptoms that are associated with ME/CFS. Of those, 11 of the gene products show a significant change in ME/CFS compared to healthy controls. And primarily those are in the glucocorticoid receptor signaling pathways, NF-kappa B signaling, and tumor necrosis factor alpha signaling pathways. So there's a number of putative treatment avenues that we're looking at trying here, one primarily being this area of tumor necrosis factor alpha. Next slide. So this is just a zoom in on that main area of the tumor necrosis factor alpha pathway. And so this involves treatments with things like infliximab, adalimumab, or etanercept. And these are generally tumor necrosis factor alpha inhibitors. And then on the next slide, I wanted to point this out because this is an interesting side finding that I'm starting to follow, at least in my line of research. So one of these misregulated or overly expressed genes in ME/CFS was something called FKBP5. And this has been shown to be increased in groups that have traumatic exposure in their past. So chronic pain after trauma may depend on this gene in specific. Now, one thing that I have been doing in Gulf War illness, because that is a military illness, there's a lot of overlap. There are a lot of individuals that are comorbid with PTSD. And we are finding that if you have comorbidity with post-traumatic stress, that your symptom presentations as well as your biological presentations are different than if you just have a Gulf War illness alone. And so this leads us to believe that there may be a similar subtyping strategy in ME/CFS, such that individuals who may have prior traumatic exposure may require a different treatment course or different treatment compared to individuals that just have ME/CFS alone. Okay, next slide. So now this is we're talking about our second study here. So this is looking at the single nucleotide polymorphisms, or SNPs. And this is looking for not gene expression changes, but changes in the actual genetic code of individuals with ME/CFS. So this was a student-run project with absolutely no funding provided. They had this idea that we wanted to do what was known as a genome-wide association study. And in order to do this, you need a large number of individuals to participate. Usually this is costly and expensive, but with the advent of platforms like 23andMe, we can get genotypes of individuals for, roughly for about $100 per individual. Now, we did not have that funding to perform a research study, but what we asked we asked for donations from patients to go and pay for themselves to have their genome typed and send us their raw data back from 23andMe. And so we opened up a website and did a large-scale online campaign to promote this. And we received, when we wrote this study, up to about 400 individuals submitting to us their genome typing. Now, since then, this has been ongoing. We haven't stopped this, and it's continually recruiting. And I believe we have over 1,000 individuals now, and it's continually ongoing. So we're getting more and more data as the time goes on. Now, we took this raw genetic data and annotated it using standard methods of CLC 138, and we focused our analysis primarily on non-synonymous and nonsense SNPs located in gene coding regions. So these are changes in your genome which actually affect the creation of proteins. Okay, so we're looking at these specific changes in genes that will affect the generation of proteins in your body. And then we compared the presentation of these changes, these SNPs within ME/CFS individuals, to a reference database. And we used for this study what's known as the Caviar reference database. In doing that, we found 5,693 SNPs that were different in ME/CFS compared to the reference group. But in order to narrow that down, we looked specifically at those SNPs that had a frequency of at least 10% difference, so either 10% in ME/CFS or 10% in the Caviar cohort. And then we performed a ratio to find where those SNPs were more prevalent. Were they more prevalent in ME/CFS, or were they more prevalent in the reference cohort? Once we found these SNPs that were unique to ME/CFS, we again ranked them on something known as the combined annotation dependent depletion score. And what this does is it ranks the gene to tell you how deleterious that single nucleotide polymorphism is. So if you have that change in your genome, it can lead to greater problems later on. So higher the C-score, the more dangerous the SNP is. And then we looked at all these SNPs that were different in ME/CFS and grouped them again into this pathway level description using the consensus path database. Next slide. So if we look at the 50 most frequent deleterious SNPs in ME/CFS and then rank them, 10 were found to have a frequency of greater than 70% or more in the ME/CFS group. So that means that they almost across the board, 3/4 or more of these individuals had these SNPs in some pattern, one way, shape, or form. And if we look at what these top 10 were, you can start to see a pattern that starts to form. So the first one here is an enzyme that's involved in the metabolism of xenobiotics in the body, so response to viruses and bacteria, which makes sense in regards to the viral component that may be involved in ME/CFS. The second one here is something that's we don't know what the function of it is, but it seems to be overly expressed in ovaries, adrenals, the central nervous system, and the brain. Again, these are all systems that are affected in ME/CFS. The next one is involved in the regulation of brain activity in mice during exercise challenges. The next one down is overly expressed in brain and the ovaries. The next one, there's no information involved in it. The following one is involved in the production of cyclic AMP, which is an energy molecule within the body in relation to MAPK signaling. Now, MAPK signaling is generated in response to stressors and immune signaling, and so this seems to fall within the general description of what you would think for someone with ME/CFS. LHB is involved in the pituitary function and the generation of sex hormone synthesis. ADAMTS19 is a possible candidate for premature ovarian failure. And then VARS2 is important for mitochondrial protein synthesis. And then finally, CPLX2 it participates in neurotransmitter release. So these together, and these are just the top 10, okay, of the 50 most deleterious SNPs. We seem to cover a lot of ground in describing a lot of these symptoms and a lot of the findings that we see in ME/CFS. It provides a coherent picture of what's going on. Now, if we take all of these findings and try and look at how all of these interact with one another, on the next slide, you'll see a global description of how these individual SNPs, these changes in your genome, can interact with one another. Now, I know this is complicated, but the way that you read this is that the little circles are gene pathways. So it's how genes work together. Now, the size of the pathway, or the size of the circle here, tells you how many of the SNPs, or how big the SNP is, or the pathway is in general. So not just necessarily the SNPs, but all the genes that are located within that pathway. So some of these pathways are larger than others, but the color tells you how well our SNPs overlap with that pathway. So the more red it is, the more of our SNPs that are located within those pathways. Now the lines are showing where pathways share genes. The thicker the thicker those lines, the more they share genes. The number of shared genes. Oh, sorry, could you go back? There we go. Yes, on this one, number 30. And then the color is the total percentage of shared genes. Now, what we did was we did a force-directed a presentation of how pathways interact. So the farther they are away from one another, the more unrelated they are. And then for those, we had clusters of pathways, and those are those circular patterns that we applied on top of those to show general pathways that work together. And the three main areas that had come up was a metabolism group, an immune group, and a hormone group. With off to the upper left-hand side, there's an additional extracellular muscular type of group that was more diverse, so it didn't get a box drawn around it. Now, in the next slide, it's going to be the exact same image, but it's going to be drawn slightly differently. And it's going to show you where those most deleterious SNPs are located. So for the most part, a lot of the SNPs that we had were found in the immune system, but we can see from the size of the circles here, which indicates the mean score of those deleterious SNPs, so how deleterious those changes are. Many of them are found, excuse me, within the hormone group. Now the color here tells you of those genes that are deleterious, which one is the most deleterious. So the darker the color means that you have a very deleterious SNP in there, whereas the size tells you the average value of of how harmful the changes are within that pathway. And so up in the immune system, we can see one here in cytokine signaling in the immune system. And just from that immune box to the upper left of that, we see something associated with inflammatory bowel disease. And then primarily all of these, the main changes, the largest number is over in that hormone group, as well as up in that diverse group in the upper left-hand corner, which is involved with extracellular matrix organization, collagen synthesis, and interactions within that cytoskeletal or extracellular system within the body. So this seems to be a promising avenue to start to hone in on certain genetic predispositions that we may find in ME/CFS. Now, of these here, we're working on a more detailed study with a larger group of individuals to look at a more statistical and rigorous approach to find out which of these individual SNPs are most definitely found with ME/CFS and may be contributing to the condition. Now, that being said, a lot of what we're seeing here seems to indicate that it may not be an individual SNP that is responsible for the illness, but a constellation of these SNPs working in unison to give rise to the overall problems that we see in ME/CFS. So not one gigantic fatal stab from one SNP, but death by 1,000 cuts by many individual small changes that are working in unison. Okay, hopefully that made sense. And then finally, we can go to the last slide. And I would like to say thank you to everyone for listening for the last 50 minutes, and I can open up the floor to questions.
Hi, Travis. This is Allison, actually, and thank you so much for such an enthralling presentation. With the type of work you do, there's great visuals as well, which is really awesome for a webinar. So thank you again. Sadie's kindly let me take the liberty of asking the first couple of questions. We had a number of questions come in. We have like a really engaged audience on. So one of them is that you mentioned comorbid PTSD that you've observed in, I believe, Gulf War illness cohorts. And which comorbidities are most interesting to you in people with ME/CFS?
Well, this one, because of my interest with Gulf War illness and the finding of how traumatic stress can alter the symptom presentation as well as biology, that's a prime interest of mine. Now, anecdotally, again, I'm not a clinician. I'm a PhD. I don't deal with patients directly, but anecdotally, speaking with clinicians, I've heard that there is a significant group of individuals with ME/CFS that may have had prior traumatic exposure, and whether that's from traumatic events in their past, such as natural disasters, that's one thing. But I'm also keenly aware that many individuals with ME/CFS have suffered traumas from dealing with the medical community in the past, who have for so long denied the existence of the illness, which can rightly so give rise to feelings of distress and stress resulting from someone not believing that you're ill, even though you are severely ill. So that alone can alter the way that the illness itself is presented. So if we think about it, that even if you didn't have this prior traumatic stress, there is a stress component associated with chronic fatigue. Now, if you drive that even further by going through some sort of traumatic event, it can alter, again, the hormone signaling, the stress axis, to give rise to an even different form of the illness. So one of the things I believe is that that chronic fatigue syndrome is not, I mean, the way it's defined right now is quite an umbrella term. And within that, I believe there are distinct subgroups or subtypes within this illness. And we do need to start to think of how can we hone in on the differences and the similarities within these subtypes. And one thing we've done is, I think people might be well aware, is that we tend to separate genders when we look at the illness, because there is a sex steroid component, so it would make sense to separate the genders. And now I'm suggesting that beyond that, we should also separate prior traumatic exposure groups.
Hi, thank you so much, Travis. We've also had a few questions come in that are sort of along the lines of collaboration and different efforts that groups have to look at heritable factors, or doing whole genome sequencing or epigenetics. And there's one in particular that someone cited in their question, which is we funded through our Ramsey program, Camille Birch and Liz Worthy at Hudson Alpha, to do whole genome sequencing on a group, and your colleague Bob Nathanson was funded to do epigenetics on immune cell subtypes. And the question was that, what are the ways in which some of these different streams of studies could be knit together? Is your group engaged in any outreach with other groups that are doing research studies of this kind? So just curious to know if you're in talks with anyone else.
Yeah, I can say that yes, we are in talks with other individuals, other well-known individuals in the ME/CFS field. I'm not sure if I'm at liberty to divulge that at this point, but we are talking with other individuals within the field, and not just within the NSU family, but others more abroad.
Fair enough. I think it's just great to hear in general that different researchers aren't just doing great efforts on their own, but that there's this kind of collaborative feeling. I think that'll get us.
I mean, we always cross-reference our findings with other individuals, and I think that the main hurdle for these genome-wide association studies, or trying to find a genetic predisposition, is that it requires a lot of individuals to participate. Now, because of that, and because of limited research funds in the area, sorry about that. It limits the number of people that can be involved. So because of this, we're working to increase our population sizes by reaching out to other groups and working with other agencies to try and expand the numbers that we can recruit.
That's great. And so, for example, for the goal of a genome-wide association study, what is the number you'd need, right? I mean, there's been GWAS studies in the past, even just looking at height and other types of factors, and they need pretty large groups. And so, what would be a target that you'd be excited about?
I think the number we're aiming for right now is about 10,000 individuals.
Mm-hmm. Yeah. That's great. And you guys are still recruiting for that 23andMe study?
Yes, we are. That's still open.
That's great. And what are some of the, a couple of people were interested in selection. You're very familiar with the field. That it's, there's some disagreement on how to define a case and how to properly characterize a person with ME/CFS that enters a study. And so one question in particular was, how do you take into account sort of current disease severity? This disease has a remitting and sort of relapsing course for many people. And so what are some of the steps you take to select your study cohorts?
So I'm going to be talking off the cuff here because I'm not involved in that end so much, but we have a rigorous questionnaire at the beginning of it, specifically for this 23andMe study. So there's certain criteria that have to be met in order for you to be enrolled in the study. You can't just donate your genome and expect it to be enrolled. One, you have to meet the criteria for, I believe it's both the Fukuda definition as well as the, and I'm going to talk off the cuff here. I think it might be the Canadian definition, but don't quote me on that because again, that's not my area. But it's two of the more rigorous definitions for ME/CFS. I think those are the ones. So you have to meet the criteria for both of those, as well as you have to certify that you have been seen by someone who's an expert in the field of ME/CFS and received an official diagnosis.
Mm-hmm.
And then they'll go through a large number of questionnaires. So the SF36, the MFI, those ones that I discussed previously, the PSPI and all of that, as well as the DePaul scale. There's several WHO scales. There's environmental scales. So it's really it's quite a lengthy process of the in order to get a description of both the individual patient's symptom presentation as well as what some of the environmental factors that may also be contributing to the illness. So not just exposure to mold and environmental toxins, but also socioeconomic class and all of these things that come within these WHO questionnaires. And by WHO I mean World Health Organization.
Mm-hmm. That's great to hear. I mean, obviously doing kind of like a deep phenotype and really understanding someone's sort of presentation is so important to the studies that you guys do. And so good to hear a little bit more about that. I know people appreciate that. And so what are some of the, limitations of the current approaches that you use, some of the bioinformatics and machine learning? And I mean, what are some of the things that you hope to see that field move toward as you improve the use of those tools?
Okay, so the main thing with a lot of these studies, so GWAS, genome-wide association studies, machine learning, and these sort of algorithms, the thing that they need is a lot of data. You need to have a large number of individuals with, and then you also have to have markers that are significant in these populations in order to start to hone in on what may be going wrong. So where we've been hampered in the past with a lot of this is small scale studies. And by small scale, I mean groups of, 30 to 50 individuals with ME/CFS compared to healthy controls in the same numbers. We can get up into the hundreds, which starts to improve resolution, but mainly for these machine learning algorithms, you need to have larger numbers than that. And so we've been working, been trying to get together to compile data from various sources that have the same sort of information and compile it into a large cohort in which we can start to run these machine learning type processes on. And one of the main things we want to do is, with this large group of individuals with these many measures of symptoms, is use that to cluster these groups into subtypes. Mm-hmm. So previous work in trying to subtype individuals has been primarily from the clinician standpoint. So what are their symptom presentations and so forth. So you may have a gastrointestinal group or a cognitive group and so forth. But this would be a more an unbiased approach in that you're letting a system looking at all of the information simultaneously weighted equally, and you let it say, this is where the individuals belong in these groups. So before, I believe the subtyping, the way I kind of look at it is that individuals were kind of categorized into predefined groups, whereas the machine learning subtyping would be, the individuals would be naturally where they are, and the machines would decide where to draw the boxes. So before it was trying to shove people into predefined boxes, and now it's trying to define boxes around the individual's descriptions. I don't know if that makes 100% sense, yeah, but that's how I view it.
It absolutely makes sense. I mean, that's what you want to see in science, right? Is these kind of unbiased approaches and taking a new look at things. And I think your group at NOVA has really embodied a lot of that. And so you've just been an exciting group to watch. We're actually at the top of the hour here. And so I think we'll need to close out, although there are many more questions that we would have loved to have asked you. Just really quickly, I'm going to give a plug to the remaining 2 presentations in our Advances In series.
Before you do that, can I just say one last thing?
Oh, of course. Yeah, of course.
Okay. Sorry, I just got an email or a text from Nancy. So I'm putting out a plea for more volunteers for this study. So if you're interested, you can contact the Institute for Neuroimmune Medicine, and we'll direct you to the right location in order to participate in the study.
Absolutely, and we'll be sure to send out a link to all of the folks that were on the webinar today, so they can, get routed to your website and see a bit more about your study. So, thank you again, Professor Craddock. It was a really great presentation. And yeah, I'll just really quickly finish this plug here. The next in our series is on November 14th with Dr. Amy Pearl. She's going to be speaking about persistent pathogens that could be driving ME/CFS. And finally, in December, we'll have a really interesting study that took a look at large insurance databases and tried to get at a better estimate of prevalence. Some of the prevalence figures that are cited are a little perhaps outdated, and so this is a look at trying to see if we could home in on an updated number, as well as some of the demographic characteristics using that data with Charmian Presquier, and that's December 12th. And just finally, if you are interested in this webinar series, we make it openly available to the community. It's a resource that we're really excited to co-produce with the researchers that join. You can take a look at what's coming up on the schedule at the link there. If you want to stay updated, feel free to sign up for our newsletter at that link there. And then finally, if you're interested in contributing, these presentations cost money to produce, and so we're always looking for folks that would want to contribute these and help make this resource even more widely available. So with that, I'll just thank you again, Professor Craddock. Thank you again for all your work and for presenting it today.
Thank you for having me.
Okay, bye-bye.
Bye-bye. Thank you.